the Dutch famine of 1944 to 1945 was a bitter phase of the Second World War. For nearly 6 months, there was a severe food crisis, especially in the northern and western parts of the country. But the effects of this historic event left a mark that lasted for generations. Around 18,000 people lost their lives and 4.5 million people were undernourished. Recent research has shown that the food shortages experienced during this period, especially by pregnant women, led to health problems such as obesity in children and genetic changes. Epigenetic changes are not direct changes in DNA sequences, but changes in the interactions of DNA within the cell. These changes occur through interventions at the molecular level, either in the DNA or in the histone proteins that surround the DNA. This ultimately affects the production of RNA from DNA, which in turn can lead to changes in protein production. Epigenetic changes can be thought of simply as chemical changes that affect gene expression. Genes are the building blocks of proteins that function in our bodies, and these proteins are synthesized based on genetic information. This process is called gene expression. Epigenetic changes that occur due to environmental factors not only affect the individuals exposed to the effects, but these changes can also be passed on to subsequent generations. This implies a more rapid and dynamic process of change compared to DNA. So what were the epigenetic consequences of the Dutch famine? The researchers focused on changes in DNA methylation, a chemical marking process, as part of the consequences of the Dutch famine. These changes occurred in genes involved in metabolism, growth, and immune system responses. The addition of methyl groups on DNA affects the protein production process. This results in an increase in methyl groups in certain regions of DNA, leading to less protein being produced from these regions. A decrease in methyl groups increases protein production. During the Dutch famine, changes in methylation in the DNA region of a gene called IGF-2 were observed in unborn babies. IGF-2 plays an important role in growth in the womb, and it is thought that these changes may be associated with problems with memory formation and growth. Another gene affected by the Dutch famine is POMC. This gene plays a critical role in appetite regulation and metabolic activities. Increased DNA methylation of the POMC gene was observed during the famine, which may lead to a decrease in proteins produced by POMC. This could mean that individuals exposed to famine may have an increased risk of obesity and develop other nutritional disorders. Finally, a gene called FADS2 was also epigenetically affected during the famine. This gene is involved in fatty acid metabolism and suggests that changes in DNA methylation during famine, passed down from generation to generation, may increase the risk of cardiovascular diseases. The studies examined not only physiological changes, but also psychological effects. They found that individuals exposed to famine in the womb or in early childhood were at higher risk of developing psychological problems later in life, such as depression, anxiety disorders, and schizophrenia. This shows that the Dutch famine was not just a historical event, but a complex one, with genetic and psychological effects across generations.